Okay, so just to say, we're also very pleased to have Cath Noakes, who many of you will know is playing a leading role in the UK's efforts about transmission of COVID. She's a member of SAGE. She's a professor and co-director at the University of Leeds and co-director of the Leeds Institute of Fluid Dynamics. We're very pleased to have you, Cath, to give us your talk on understanding COVID-19 transmission mechanisms and uncertainties. So over to you.
Okay, thanks, Paul. Yeah, I see I've just now lost half an hour of writing my slides time, but I'm sure it'll all be fine. So I want to talk, I guess some of this is overview, some of this you've probably covered already, but I really want to think about some of the challenges with the uncertainties within how we understand transmission. So starting from the basic principle, I mean, we can see straight away, just considering how the disease is transmitted, that we've got multiple opportunities for complexity and uncertainty in here. So we know that the majority of transmission will be through respiratory aerosols or respiratory secretions. It is possible that there is some transmission through fecal matter as well, but I've not shown this on this diagram. But even that respiratory activity results in a whole raft of different sizes of aerosols, from submicron up to well over 100 micron in size. And of course, I think we missed the bit that we have called secretions here, but that simple act of you cough in your hand or you wipe your nose with your hand, and then you've got virus on your hand, so we can transfer that onto surfaces. And then once with those different sizes, there are then multiple potential routes that a susceptible person can become infected. And we've tended to largely group these into three, although there's obviously complexity within these. So airborne is, in this case, is referring to greater than 2 meters away in a shared room, which obviously, as you well know, the 2 meters is a nominal 2 meters, but at a distance, shall we say. We've put close range, which actually groups together aerosols and droplets. So a proportion of close range transmission is airborne. Some of it is probably also larger ballistic droplets. The proportion of which these are is really uncertain. And then obviously the via contans and surfaces as the third route of transmission. But within all of these, there's an awful lot of uncertainty. I thought it was worth touching on how do we get some of that evidence for transmission. And I've got quite a big list here of different approaches for collecting information on transmission. And I think the first thing is there's no one defined way of measuring transmission and understanding it. And what we're trying to do is triangulate data from lots of different sources to infer the right information and recognise the limitations with that. So, for example, we know that where animal studies provide us some useful information on transmission. We know from that both aerosols and fomite routes are possible. It's hard to say what distance that can travel yet, because most of those setups are quite small distance. But we also know that hamsters are not people, and hamsters don't cough and blow their noses like people. So there's a limit to what we can get from those, although it gives us some understanding of the potential mechanisms. I think one of the really big sources of evidence is outbreak data that allows us over time to start to pick out common features and common risk factors. And if you think back over 12 months, you know, we were starting to say, oh, there might be airborne in some of these, one or two of these super spreading events, but of course at that time there's only maybe 1 or 2 reported in the world. It's very hard to work it out from very small numbers. You have to almost let that body of evidence build up to an extent to really understand where transmission might be happening. But I think what you can see from these outbreaks is a set of common features now. We know that where people have close interactions together, where they're often, you know, smaller spaces, workplaces, together in or restaurants, or then there's definitely opportunity for transmission. How that transmission happens is not always clear, because there's again multiple opportunities for different routes in most of these outbreaks. But then when you start looking at the super spreading data, they're quite interesting because nearly every one of them is indoors. A large number of them are associated with poor ventilation and associated with also with higher aerosol generating activities, so things like singing or aerobic activity. And nearly all of these are where you've got people who are not wearing masks. Of course, you can't just draw conclusions from one activity though. I think the contact tracing data is quite interesting. Contact tracing data was never set up to be an epidemiological study of how virus transmits, but it allows us to get some clues, and it has a limitation, and it is only as good as the data that is reported. But you do see that when people do report, that there is a higher number of contacts at 1 metre than at 2 metres in that data. So you can see that there is some proximity effect going on there. Again, it doesn't tell us the exact route of transmission. I think looking at where the locations and the types of venues where things happen, again, gives us some clues. So we know that there's very little evidence for transmission outdoors, but there is, apart from, where people are very closely mingling, things like parties, but nearly all of the transmission happens indoors. And we can start to pick out venues and activities that potentially have these higher risk factors. So some of those, perhaps the gyms, hospitality, some of the bars and things like that. One of the other interesting things is where there is evidence of something and where there isn't evidence of something. So I think the ventilation aspect is interesting here. So where we've got super spreading events, they tend to be associated with very poor ventilation. Things like some of the hotel quarantines data that's starting to appear now, again, you can see evidence of poor ventilation and pressure relationships between spaces. But where I haven't seen any evidence yet, which is quite interesting, is transmission in very well ventilated spaces or transmission throughout a recirculating ventilation system in a whole building. And that's not to say it doesn't happen, but we haven't seen the evidence of that appear yet in the literature. I think as we've got more sequencing of virus, we're finding that helps us connect things together. In a country with very high prevalence and very high numbers of cases, it's actually very hard to unpick what happens. But once you get lower numbers of cases, you can start to be able to connect the data together, and that's really helped, particularly in some of the New Zealand cases where they've got very small numbers and they can be really quite certain who transmitted to who. We can start to do some more detailed intervention. We can do case control studies to look at how people who have got infected compared to people who haven't got infected by matching groups of people together. There's always biases in that though. You have to be a little bit careful that your control group is often biased. And we can do cohort studies. That requires quite a lot of effort because you have to basically follow that cohort of people. I think the population data tells us some things. It tells us about major interventions, so it tells us whether, if we have a lockdown or we close schools or whatever, that the impact of those. It doesn't tell us very much about any of the environmental mitigations, and even things like mask mandates. It's very hard to unpick that from population level data. Paul just touched on his introduction there about environmental sampling. I think this gives us some clues, but we also have to be aware that it is actually quite hard to sample from the environment. It's much harder to sample from the air than a surface. And of course, in the air, you will only find the virus if the infected person is present and emitting virus or emitted it very shortly before you sampled. So if, for example, somebody got infected in a supermarket and was caught through test and trace, if you went back to that supermarket to sample for virus, you would not find it in the air because they were infected 5 days earlier and it just will not be there. And then I think one of the other major influences is about the physics of aerosols and microorganisms and the biology of this as well. We are talking a lot about this. This does not tell us how transmission happens, but it helps us to understand the other evidence that we've got, and we can match the two up together. But in itself, it's not proof of transmission, it's just proof of principle that it could happen. And a touch about uncertainties and thinking about this transmission chain. So if we start at the beginning with the infected person emitting virus, There are multiple factors which affect this, the viral load, the size range of the aerosol and their activities, together with whether they're wearing a mask or not and the particular hygiene behaviours. And I've underlined 3 there because they are ones where we have a huge amount of uncertainty. I think, strangely, viral load, I think there is, I wouldn't call it uncertainty, I think I'd just call it a very big range. So we know that this can vary over probably 8 orders of magnitude or more. And there's some reasonable data now that shows that. I think what we don't know is how frequently those different viral loads occur and what that might mean for the emission rate. We've got some reasonable data on aerosol sizes that people produce, but obviously that we don't have it for all activities, and we don't necessarily have it for the infected, for the actual virus itself, which is a big gap in our knowledge at the moment. When it comes to the environment, we see multiple factors in here which will influence the transmission, including the ventilation, distancing, whether the virus survives, which is influenced by the temperature and humidity, and then things like the activities around cleaning that you might do in that environment. And then when it comes to exposure, your exposure is controlled by some very personal factors around the hand hygiene that you practice, the whether you wear a mask, whether you touch your face frequently, and how you your breathing rate. But then once you are exposed to it, what is the efficiency of that virus being deposited into the respiratory tract? Does it vary with different routes? And how much virus do you need to get infected? And I think we should talk about dose response here rather than infectious dose, because I think there's a spectrum in here. And again, I've underlined those 2 because I think that's where there are some really big gaps in knowledge. I think the ones which aren't underlined, we have a lot of uncertainties in them, and there's a lot of gaps, but they're probably less impactive on the transmission route and our understanding of transmission than the ones that are underlined. And the whole thing of this is a combination of trying to figure together the behaviour, the physics and the biology of this transmission. So to just illustrate some of these a little more, and I suspect you've probably looked at some of these over the course of the past day or so, but we know that the virus is very small, 100 nanometers, but it's not on its own. It's going to be within respiratory fluids. So therefore we need to consider the properties of those fluids and how they might evaporate and how they might behave. We do know you get very rapid evaporation to a final diameter. What we don't quite know yet is actually what this does to virus survival, and we suspect you get a very rapid decay in virus survival as well, followed by a longer, slower decay. And we also know that we get different different aerosol production under different activities. The graph on the right is from Johnson et al. 2011 paper, and I think it's quite interesting to see that the different models show slightly different things. I think there are some questions as to how much of what we see here is what really is there, and how much is an artifact of how those various studies have measured the different sizes of aerosols and droplets from somebody's exhaled breath. I think we also know a huge variability in the aerosol generation from different people, and these are just particle number concentrations from healthy volunteers under different activities. I think what's quite noticeable is when people are singing or speaking, the data is quite clustered, or more clustered, and you can see a clear increase with the volume of the sound. But when you look at where people are breathing, there's a huge variability in the numbers of particles that different people produce just simply by breathing. So we need to make sure we can capture this variation. You know, there's 2 or 3 orders of magnitude difference there just from an individual. And then the same with a cough. And some people will breathe out as much as somebody else might cough. And of course, we don't know what this translates to in terms of actual virus yet. So, I mean, this is data for influenza. We know that this is not influenza, and it probably does behave slightly differently. But I think, again, the important thing here is this is data from multiple volunteers with influenza, and you can see that the amount of virus they breathe out is again over about 4 orders of magnitude. So we get some really big variations between individuals. And you can't just look at somebody in the street and know which one is going to be the high producer and which one is going to produce almost nothing. So we might find that some people just are barely able to transmit it because they don't produce enough, whereas others are teeming with it and can produce loads of virus. Again, I mentioned viral load, and I think again you probably talked about this, but there's a couple of good data sets now on viral load and how it varies with time. The one on the left actually has just been published, so that data set's quite useful. But we know again that we've got quite a big distribution, several orders of magnitude of viral load. And we know that you get different results if you've got a nose swab and a throat swab as well. So it's not always consistent. We also know that sometimes you might take a nose swab and you get a high load. You might take a swab from the same person 2 hours later, you'll get a different number. So it's really uncertain. I think quite a lot of it will depend on how that swab is taken. And then we also know that people are most infectious at the very beginning, probably if they do show symptoms, they are at the point where the symptoms just start to appear or just before that point of symptoms, which is obviously one of the things that makes it very difficult, not only to manage transmission, but also to manage the fact that we very rarely sample people at the right point. So for example, if you look at the CT values, so the infectious viral load in the test and trace data, it all shows a lower viral load than many of these other papers do. And the reason being is that those are the people who have got symptoms and thought, oh, I better go and get a test. And we've missed day 0. We're probably measuring them at day 2 or 3. I think, again, there'll be a lot of discussion about this in this group, but the complexities of our exhalation physics, there's a lot we do understand, a lot we still don't understand. And I think a lot of one of the big questions is, how do we translate some of this variability and into real life risk models? Because I think, we can't just assume that people are always breathing out a cone of a higher concentration cone of virus. We need to think about how do we incorporate some of these short duration events like these violent ejection, turbulent puff type coughs into the sort of well-mixed models that many people are using at the moment. And I think many of you will have seen this picture before, but thinking about how we've got this really complex behavior going on very close to a source, in that you are exposed to a whole range of different sizes of aerosols. They will behave differently in terms of their physics, but they will also behave differently in terms of where they can actually penetrate the respiratory system and deposit. And if we were worried about tuberculosis, we could get rid of all of the thoracic aerosols and nasopharyngeal aerosols because they can't penetrate the deep lungs, so they don't matter. But unfortunately for this disease, all of those sizes probably matter. I think a lot of people have been using these types of mass balance models of varying levels of complexity. I think they give us a really good initial understanding of the factors that matter and the relative importance of different factors. But I think we are now reaching the point where we need to start thinking through the next level of these, and how do we enable these models to incorporate some more of the complexities of the flow physics that happen in real rooms, rather than just simply stating that it's a well-mixed space. And I think some of you may have seen that there was quite a lot of media attention to a study in PNAS where the reporting was around 6 feet is no different to 60 feet. From a well-mixed model perspective, no, it isn't. I think many of us might think, is it really that well mixed that we're certain about that? So I think we ought to be thinking a bit more about the mixing and problems. But we can, even with a well-mixed model like that, use this to look at some relative effects And I think the relative effects are quite interesting, because even within a simple well-mixed model, if we then incorporate the fact that we've got a different size of a space, a different ventilation rate in a space, different aerosol emissions from vocalization, and different durations of exposures, we can see that we get some relative risk factors that vary quite significantly. And in this particular model here, you can see that, for example, the supermarket comes out very low, and that's really simply on the basis it's a huge space, people are in there for a short period of time, and people are passive and wearing a mask. Whereas, you know, the choir outbreak on the towards the right hand side is much higher, because we're now in a smaller space, poorly ventilated space, longer period of time, and higher aerosol generation. So we can look at, even with these simple models, look at the relative order of magnitude of some of these different risk factors. I do want to talk about how we link exposure to risk of infection, and many people will have used the Wells-Riley model. So Wells-Riley has a lot of advantages in that it's a very simple model, and the version I've shown there is the steady state version of that. There is a non-steady state version as well, based on the Gametonian-Ucci formulation of it. It is a dose response model though. It's an exponential dose response. Essentially it's assuming it's the ID60, so the 63% infectious dose, but it basically incorporates the dose response within the concentration model. But if we look at dose response, we can look at modeling exposure and then use a dose response curve. What we don't have yet is dose response for SARS-CoV-2. This plot just shows two of the other coronaviruses, SARS-CoV-1 and HCOV-229E. And what you can see from these is there is a big difference between the two. There is an order of magnitude difference between the two. Is that in part will be to do with differences between the infectivity of those, but it may also be to do with the difference between how they're measured, whether an animal or human study. Just to touch on quanta, and we know that there are huge variabilities, and this is data from other diseases. We know that there are variations from many other diseases too, particularly influenza has several orders of magnitude difference on it. And then for SARS-CoV-2, some estimates from the Buonanno paper, which many people have used already. Again, we can see we've got variabilities ranging over about 4 orders of magnitude, depending on the activity and the viral load in exhaled breath. And then we can start to put these together with what happens if we've got these in a different ventilation rate with different quanta, whether it's in transient or steady state mode, and the time that you might spend in a venue. And again, you can see some really big variations in predicted risk simply by varying these parameters. And all of these parameters are uncertain. Most of these settings we might just be picking, you know, where do we pick in here? I'm going to touch on, some of you have seen the Skagit choir models already, but the case that this was the scenario where we've got a very high secondary cat rate amongst attendees at a choir. I think what's quite important is that there was no clear evidence of proximity in this. The cases were dispersed through the room. And this was modelled through a model group I was involved in, but led by Shelley Miller, which looked at using a transient model, a transient Wells-Riley model with a Monte Carlo approach to estimate the quanta, and looked at the variations in these different parameters to understand the likely risks. And just to show some of the results from that, the graph on the left, the loss rate coefficient essentially is the ventilation rate, but includes deposition and other losses too. And we can see that this is with the predicted quanta generation rate of 970 quanta per hour. But even that has quite a big band. What we might predict the effect of increased ventilation might be. And then on the right, we're looking at the duration of exposure. So the actual event was 2.5 hours, but if we reduce that duration down to say half an hour at a much higher air change rate, we might well then see a significant reduction in the likelihood of infection in that case. And again, just to show some of the variability in this, this is a model we put together earlier in the year where we modelled the potential benefits of different types of masks, and looked at different types of mask materials and took the data on the efficacy of those mask materials for some data on the measured RNA copies in room air. We assumed whether different proportion of aerosols were infected or not, and looked at over time and what the infection risk would be through a dose response curve. And I think the key thing to just highlight in here is the variation in these, the ranges. Some of these have very long tails on them, reflecting some of that uncertainty. And I've just realised that it's missing the scale off there, and I can't remember off the top of my head what the scale would be. So apologies for that. Last one I just want to mention is hands. We are focusing quite a lot here on aerosols, and there's a lot of talk about aerosols and ventilation. I think we shouldn't discount hands. I suspect they're not the major driver, but I suspect in certain settings there's quite a big opportunity for transmission through fomites. And in a way, this is far more complex than the air because the sheer number of parameters that are involved. You know, if you think through the process of how you might transfer a virus from a surface to your face, you know, the virus has to have been on the surface in the first place. You've then got to touch that surface. You've got the area of which you touch, the frequency with which you touch it, how efficient that virus transfers onto your hand, which might depend on whether it's wet or dry, and how much was there in the first place. You then think about how frequently you touch your face and which bits of your face you touch, because you've got to get it to the mucous membranes, and how efficient that transfer is. And again, what the area and the number of viruses that get transferred. And then, of course, the mitigations with hand cleaning and decay rates on those surfaces and on your hands. So a lot of parameters. Again, we can do some modelling, and this is just quite a simple model of looking at how the variability in viral copies. What we've assumed is as a surface is inoculated, and then whether somebody touches it immediately or touches it up to 48 hours later, and it will depend on the decay on that surface and the whole raft of different hand touch, face parameters and viral transfer data. And we're lacking a lot of data on viral transfers and a lot of data on the behaviour of people touching their faces. So just to conclude, I think there are some things that we're reasonably certain about. I think we're pretty confident now that we know that at close range you've got the most exposure to the most virus, and in many settings that is going to mean that that's the highest risk that people face. You are probably more likely to be infected from a friend than you are from a stranger, and you are more likely to be close to a friend. We do know that far-field aerosols matter, particularly for the longer duration exposures. So it's probably fairly low risk for short duration, very short duration, but longer durations, an hour or more, it probably matters. And we don't know how much it matters and even how many of those we missed. And I think we shouldn't discount the surfaces, particularly if you're sharing that space with somebody infectious, like a household or a workplace where you're in a close space together. But there's a lot of gaps in here, so we particularly need more information on the viral load and then the size of the aerosols that contain the virus. So we at the moment are making quite a lot of assumptions that we can just distribute this virus by volume across particles, and that might not be the case. We don't know that for sure. And then the data on dose response is really quite uncertain, and whether that changes with the different route of transmission. I think thinking about modelling, being able to incorporate these uncertainties into models, and quite a few of the risk models do this quite well now, but I think there's an interesting question where we're looking at airflow models and how do we bring in that stochastic element into something that's actually conventionally quite deterministic, and how do we build those, you know, look at running multiple models and looking at model, you know, in different ways. And then I think a really big factor, which is quite a difficult one to bring into the physics models, but absolutely critical, is the behavioural factors. So, you know, when we set up a model, we have to make a whole raft of assumptions, and we usually assume that people are in a particular position and probably behaving themselves. But as well as Paul mentioned earlier, how do people influence the flows when they walk and they wake? Actually, where do people spend time? And can we assume that they did spend their time nicely distanced apart in an office, or actually did they all go and have a cigarette break together in close quarters, or did they all go and share a vehicle together? So we have to be really quite careful that we can consider these different factors in there. And just to finish off, a few thank yous, because obviously a lot of the work I've shown here is not my work, it's from the whole raft of other people I've worked with over the year, including many people who are in this call. And thank you to all the people who've been working on this so hard over the past year. And I will finish there because I'm over my time.
Thank you very much, Cath. That's a fascinating talk. Thanks. And I agree that these uncertainties really make it very difficult. We are slightly running behind time, but I just would like to put one of the questions to you, if you don't mind. And from someone anonymous, it says, what factors make different respiratory pathogens more or less prone to different transmission routes? For example, do we know why foam-like transmission is so much less important for COVID-19 than some other diseases?
That's a very good question. I don't know if we do know it's less important or more important, or perhaps I should say we don't know if it's more important for the other diseases. I think there's a whole raft of different factors. Some of it will be whether the virus or bacteria is aerosolized in a particular way, so whether people produce that aerosol. Some of it will be to do with the receptors. So for example, tuberculosis, the receptors are the alveoli of the lungs, so you have to get it there. Whereas SARS-CoV-2, these ACE receptors are all the way through the whole of the respiratory tract. So in a sense, you can infect somebody in their nose as well as in their lungs. And so I think that is, and then of course many diseases that fomite transmission is, is opportunist pathogens. So for example, if you look at things like MRSA or Pseudomonas in a hospital, a lot of those are, it's the transfer of the virus, the bacteria into a wound site or into a, rather than as an inhalation. I do think that we are, for respiratory pathogens, I think we'll find that more of the ones we thought were on surfaces are also in the air and are transmitted in similar ways as we go forward.
All right, thank you very much. I'm afraid we don't have time for any more questions. Thanks very much, Cath.